Welcome to Entangled Health, where we explore the intersections of quantum science, AI, biotechnology, and human well-being. My name is Thomas, and I'm convinced that health innovation happens at the entangled boundaries, where cutting-edge physics meets biology, where consciousness research intersects with technology, and where visionary thinking transforms into practical solutions. We bring together pioneers, researchers, and innovators who are reshaping how we understand and experience health. My guest today is Samarth Sandeep, CEO and co-founder of If and Only If Technologies, quantum computing pioneer and board co-chair of the California Native Garden Foundation. Samarth's journey, which we will hear about today, represents the kind of entangled thinking this podcast celebrates, where environmental stewardship, technological innovation, and healthcare advancement converge. We will explore how his quantum-enhanced biomolecular modeling is revolutionizing drug discovery, especially in the development of biologic therapeutics and biomarkers, his vision for quantum network solutions providing meaningful improvements in the biopharma and agricultural industries, and why his path from quantum algorithms to green finance reveals the interconnected nature of our health challenges. A quick disclaimer, all chats at Entangled Health are a co-creation between the guest and myself, and not an official statement from our industries. We are here in a personal capacity, so everything we share is just our own take, not the official view of any company, nor of my employer or any other organization. To add a little bit of dopamine, later in the show we'll dive into the Entangled Health question pool, where curiosity lives in Hilbert space. And here's how this works. 1. You submit a question via LinkedIn or episode comments. 1 per listener, keep it spicy. 2. A quantum algorithm running on real hardware picks which one we answer. And 3rd, our guests add their own question to the pool and then answered it live. The rules for now, it's quite simple. Ask anything. The guest might decline, but where's the fun in playing safe? And if you reach 1 million listeners, we might change the rules, but that's a bit far out. Stay tuned, and who knows what the quantum dice will reveal. So finally, the intro is over. It's always cool to see the guests and waiting till the intro is over. Yeah, Samarth, really hello to the show and good to have you today. Thanks for being my guest. I think we start with the first question that I always ask, making you the 4th one, so making it a routine now. When we first discussed this interview and you heard Entangled Health, what came to your mind? And I mean, how does this concept resonate with your work, bridging quantum computing and stuff?
Awesome. Well, yeah, that was a very fun intro. And thank you so much for having me, Thomas. Definitely. I think for me, Entangled Health, the entangled part is where I really think about quantum. So not just quantum in terms of course, recently everyone's been talking about quantum computing, where we'll have some type of quantum device that can do some type of quantum algorithm, but also quantum information science as well as quantum chemistry, and especially the quantum chemistry behind life. That's something that is still being well understood, and so the healthcare impacts of that understanding of those subatomic interactions in chemical systems that are part of our biological systems. That's definitely something that, when we say entangled health, that's sort of where my mind goes immediately in terms of entanglement and whatnot.
This is cool. So thanks for sharing this. Our listeners, if we have some, just kidding, come from diverse backgrounds. I mean, that's the idea of the Entangled Health, to really bring together a lot of different disciplines. And I think it's always interesting to hear individual journeys. I mean, what brought us here where we are now. So, and I think when we had a brief pre-discussion, so your story is really connected. I mean, interconnected, from quantum consulting, Deloitte, to, I guess you will talk about this native plant installation, non-profit, pioneering quantum enhanced bioinfo with a focus on economics, if I'm not wrong. Can you just walk us through with your own words? I think that might be better than me explaining your CV.
Yeah. No, I appreciate that, and I think you covered like some of the major points.
Yeah.
But yeah, basically, so I've been sort of interested in biomaterials since I was in high school. I think my interest was sort of different than most people. I think most people, when they're interested in biomaterials, they're interested in it for developing maybe different types of packaging or that kind of stuff. My interest was when I was in high school, I was trying to build a seawater powered car. And my interest was how can I better generate hydrogen gas to be able to turn the seawater into meaningful energy that can then be used to power the car. And that's what led me to learning about some research on how bacterial rhodopsin was being engineered for basically helping in the hydrogen generation process. And that fascinated me. And I was like, okay, this is something that I will probably not understand in high school. I need to go to school and learn about this. So that's why I majored in materials engineering at UC Davis.
Yeah.
And then it was here that I happened to, I had, it was in starting this company that I was able to go to Y Combinator startup school. And the one that I went to in 2016 was mostly very boring because they were just talking about you need to grow your customer growth by this much and like how do you do like your like pro forma and whatnot. It was like mostly startup stuff like that, useful stuff, but for me, like as a kid, I was like, it's boring. But there was one presentation that was very interesting, which was Chad Rigetti talking about why quantum computing was something that was going to be super important and helpful and interesting in the future, especially for new material development. So this has stuck with me for a while, and then I kind of had forgotten about it. And then Rigetti was doing a hackathon at their office in Berkeley. So then I was like, great. Okay.
You've been in this office with so Rigetti's. Oh, okay, cool. Okay, no, let's.
Yeah. Well, it was great because like this was when they still had, I think their office is still like this, but they had like the device. They just have the device in like one corner, and they have like all the rest of the tables in the break room and stuff like right next to it.
Okay.
So yeah, no, it was. I think I don't know if they did another one after that, but this was in 2018, and I went with some of my friends, and we were trying to look at how we could do modeling of. At the time, like I had no idea how to really write quantum algorithms, or I really understood the space very much. So I was just like, okay, can we do like quantum, a sort of QAOA approach of looking at brain scans in some way? I don't understand why I thought that was interesting, but it appeared like something that could be useful.
Yeah.
But then that didn't really work. We didn't do very well, but I was definitely curious, and I learned, at least during the hackathon, about how one could write quantum algorithms, and also how there are devices that exist where you can actually run them, and potentially there's the combination that will get you some type of advantage in modeling some type of biological systems. So that's when I decided later that year that, I'm going to start my own company that's going to be focused on biological modeling. I think the main thing that I was interested in was I wanted to see if there were improvements that could be made in protein docking to start with. So I had heard about a professor at UC Davis who was looking at how yeast proteins, or proteins, specifically extracellular proteins in yeast, could bind to nutraceuticals, so things like curcumin or whatnot. So that there could be like some sort of higher quality dough that could be basically made from this, and they could sell it as some type of supplement. So I was like, I sort of had worked with them on, okay, how can we make docking better? And that's where we looked at protein pruning as a way to identify binding sites before, or really blocking atoms that are blocking interesting binding sites before doing docking. And then did that. That was okay, but really what took off the company and made it a company rather than just a fun set of projects was we wrote about protein binding with regards to hydroxychloroquine and azithromycin, and how really like we were at the time, it was obviously clear. So this was the beginning of the COVID pandemic, and it was clear that these drugs don't really do anything. But there was, or like at least like potentially the combination did something. So there was a good review on azithromycin maybe doing something, because it's already been used in lung health for a long time. But at least that combination was something that was just kind of like thrown out in the air. So we wanted to just really have a molecular model to prove that it didn't really do anything, so that it did not keep getting airtime. So that's when we were looking at, okay, can we just do some type of docking with the spike, SARS-CoV-2 spike and ACE2, which is its known receptor, and then being able to see like what are these atoms, or are these chemicals, hydroxychloroquine and azithromycin, doing on this active site. So we basically showed it does not do anything. But that paper got cited a lot because it was something that people were needing at the time. So that's how we got our first client at UC Davis Medical Center, and with my company If and Only If Technologies. And so I was working on that for a good few years in terms of, and that's when I think we first met, when we were part of the Q Farm group. And
now we have it. So we need to record this podcast.
So at that time, when we met, we had just sort of wrapped up developing this our version of the quantum approximate optimization algorithm, QAOA, for MaxCut, but specifically for proteins prior to docking. And that was something that I had come to you and said, hey, was this something that you would be interested in as a product? And since then, so this was like, I think 2020, 2021. But since then, we have continued working with our collaborator at UC Davis. We found other biopharma startups, as well as other large biopharma companies as collaborators. As well as research hospitals, and in working with them, we have created a consulting practice as well as software development studio, wherein we develop other quantum algorithmic approaches, especially looking into ways in which we can start to apply quantum chemistry and looking at biologic systems. So we have everything from, the way we like to say is we can do everything from if you have not a lot of time, we can look at docking, which is not a very good method, but it's a very quick method for understanding how proteins are interacting. Especially our focus is on large, like multi-thousand atom proteins and how they interact, which is something that most docking solutions do not really get into. And then all the way to doing full molecular dynamics, where we would be able to actually see, okay, given some time series, what's the actual interaction that's happening, especially looking at the orbital level interactions. And then using these insights to then actually rank literature, rank clinical trials. That's sort of been our interest. It's like, how can we use biochemical insights to drive this kind of interaction?
Before we go even deeper in the tech stuff, so this was after Deloitte, right? Or was it in parallel to, because I'm
this is actually before. So I did this for until 2023, working just completely on the startup, and then I saw this opportunity from Deloitte where they were looking for people who had similar background. I was like, this is interesting. I would love to see if I could help with this team. So then I joined Deloitte in January of 2023, and initially I was told that we're going to be working a lot on life sciences and chemistry and that kind of stuff. And then their belief, and I mean, some people's belief in the field as well, is that this is something that is not a near-term use case, whereas finance and other areas are more of a near-term use case. So I was helping more on projects like that. I did end up writing one paper with a team at IBM, on quantum chemistry based, well, really more like it wasn't quantum chemistry, but molecular properties, material science properties that could be derived with some type of quantum graph neural network, which was interesting because I had never really focused on machine learning based, quantum machine learning based approaches. But then, yeah, I ended up leaving there August of 24 because yeah, I think I did not really fit with my background more in simulation, and doing that kind of work didn't really fit with their long term goals. And then since then, I've been working back on the startup, and it was
a bit of an
period.
on-off stuff.
Yeah. So it was in between that period that, like, at Deloitte, wherein I found this garden. It was close to. So I used to work a lot out of the Deloitte San Jose office, and that office was very close to this garden, and I sort of walked past it a few times. This red fenced building with all sorts of plants, and because I worked at like this finance company, and I had sort of heard about colleagues of mine that were working on green bonds and that kind of stuff, and outside of the quantum chemistry, especially in health focus, that was definitely my interest, and still is, in terms of finance and especially new models of finance. So when I heard that this garden existed and that people in my firm were looking at that kind of stuff at Deloitte, I thought, perfect, I'll connect these people together and we'll see what can happen from there. And then I got on the board, and I'm still working there now as the co-chair of the board. So that's I guess in summary, basically how I got from where I started with starting my company to now working on
finance,
this together.
So and this is, I mean, this is really fascinating. So you went into quantum for COVID. I mean, like, everyone was busy with COVID last with these masks, etc. Then you jumped on the consultancy, and then you identified nature, and then so with this garden, you decided to maybe go deeper into things that are not necessarily only finance. Is this true? Or, I mean, just asking suggestive question. But so did the, let's say, the interest in the garden transform some way you were looking at solving your problems, or not? I don't know. So just asking.
Absolutely. No, I mean, I think, so of course, part of my interest was, like, I think that the thing that guided it a lot at the time was the finance aspect of things. But definitely, why I was interested in the place in the first place was, I think last, in our pre-discussion, we had talked a little bit about the community garden that you have access to, right? For example, and most community gardens that I think people have access to, where generally there's maybe some plot, and usually it belongs to some community association or something, and different people in the neighborhood basically own different parts of that plot, and then they can grow whatever they want in their part of the plot, and usually they grow food plants or something like that. So this is usually like what these sort of urban gardens, or I think now they're calling it urban farms, do. And even in San Jose and here in California, that's what they mostly do. But this place is focused only on native plants. That's what was really
endemic. You mean, so native in the sense of being born there?
Yes, exactly. Native and also at least non-invasive. So ones that very well work with the native plants, such that they're not like stealing nutrients and whatnot, and they're able to coexist in some meaningful way. So from the minute you walk into this space, you'll notice that actually, like, it feels more like walking into a sort of urban forest than it feels like walking into like what you would expect with of an urban farm, with where usually there's like row crops and that kind of stuff, right?
Okay.
So what was interesting to me, and still is interesting to me, is this approach of, okay, like does this sort of native agriculture lead to improvements in soil health in some meaningful ways, and then also are there some native plant species that we, especially here in California, that due to the ills of colonization and genocide of the native peoples and whatnot, we just don't have a really good grasp and understanding of. So I think one of the moments that really inspired me on this, the first moment that I saw at this garden was, I think one of the days that I started volunteering there. It was raining very hard, and we had to go in. Like there's like an inside space there, so we were just working inside. But what we were doing was we were shelling acorns. And the reason why we were shelling these acorns, so these were acorn nuts from the bay laurel tree. This is a tree that's, if you've ever been to the San Francisco Bay Area, is very, I'm not really as much on the peninsula, San Francisco side, but very big on the East Bay side. You see everywhere there. But we were shelling these acorns, and I asked the founder of the garden, Alri, why are we doing this? And she said, well, you can actually make a coffee out of this. Like it actually has caffeinated content, and I couldn't believe it because yeah, the way we were told is that the only coffee comes from the coffee plant, and it only grows in certain regions, and has this whole supply chain behind it. So yeah, there are plants like that that exist in California. I think the terminology, people who study, especially the Ohlone culture in the Bay Area, they're the sort of name for the grouping of indigenous peoples that grew up here, is that there was sort of a Garden of Eden that existed here with all sorts of plants.
I have no doubt.
Yeah. So I mean, they would, like the stories that I keep hearing from, especially members of the tribe that exist today that are retelling stories of their ancestors, is they would basically feast for days, especially during summertime, on like not only the animal bounty. They were getting a lot of oysters and a lot of other fish sources and whatnot by being on the bay, but also the variety of plants that they had. So anyways, all this to say that part of me, and one of the interests I have currently, is there probably do exist native California plant species that maybe not only have great agricultural and culinary use cases, but also potentially can provide active pharmaceutical ingredients.
Yes.
Or maybe did in the past, and can be used as the basis for developing especially new biologic therapeutics. So that's definitely something that, as in my role now as the board co-chair, is something that I'm trying to explore with some bioinformatics students, especially San Jose State University Bioinformatics Club and others. Because that is definitely of interest. I guess going back to what are the type of agricultural changes we're seeing that were interesting, that I think a bioinformatics perspective may be helpful in terms of studying. One of the things that people have noticed in the garden is that the amount of there was a sampling done when in comparing the soil that grows at our farm to others in we're part of this group called the Urban Growers Network, so like other groups of farms in the region in San Jose, and we found that the microplastic content in our soil was much less than that in others. So that's something that I think understanding why that exists, why the if there's some combination of maybe bacterial species, but also just generally using less plastic-based resources to start with, just allowing for that kind of better microplastic species would be super helpful, especially in understanding.
I mean, this is super interesting because I tend to get more and more every interview I do. The more interesting I'm getting in all these soil topics and the role of mycelium, for example, and the role of all this then back to the bacteria and soil health. So yeah. I think that this might be also good machine learning problem for no, but I think the fact that you discovered that once you reanimate the original native setting, it stimulates stuff that we just maybe can't explain. I mean, why should there be less plastic in? But in principle, there's a lot of pathways that are worth exploring. And just, I mean, always you have data, and then you try to make sense of it, and you discover new things. That's cool. This is really cool.
Yeah.
And I think then in the pre-discussion, I love that you then have via this UC Davis, or you had this idea about these smart homes or homes that can detect health issues. Or what? Could you tell me more about this one?
Oh yeah, sure. So that was not a project that we were working on directly, but
okay.
there was a team from the city of Berkeley. They were trying to. Okay. They have worked on this project called the Step Up housing with a group called BOSS, and their goal is that they're not just trying to make better housing for previously homeless people, but they also want to be able to use it as a way to help with the health issues of the homeless people. So why we got involved with them, or what we were interested in doing, and we still are, is seeing if our tool for looking at clinical trials for different patient groups is something that their doctors would be, who work within that facility, would be interested in looking at for the patients in that group.
Okay.
Especially because the reason, so we had developed this. I think I talked a little bit about how we're interested at if in terms of looking at how we can drive these biochemical insights in clinical orientation. So one of the ways in which we were trying to do this is look at protein interactions as a way for then understanding what are the causes of certain diseases, and then also what are the sort of intersection points between different diseases. Especially, I think what we were interested in was the intersection point, for example, between SARS-CoV-2 and certain cancers, as well as, so I think specifically we're looking at bulk lymphoma as one of them, and also T-cell lymphoma as another, because these are very rare cancers that already affect whoever it affects it very badly. So they try to see the impact of SARS-CoV-2 as well, and see what meaningful clinical trials exist that help these patients, since they really don't exist current trials for them right now. So that is what led us to developing this platform with the doctor at Mount Sinai Medical Center, and that tool is what I was hoping that the people and I'm still hoping that people at Step Up would be interested in, because that would allow for them to, especially if they have this kind of immuno-oncological effect, which unfortunately is only increasing by the day, that the number of people who have those kind of effects, they'd be able to find some meaningful trials that already exist, but maybe have not targeted them specifically, because they've not specifically by word found that they are part of that group, but maybe by protein interaction network, it would appear that, okay, there's actually some overlap here.
I think what we can do is, if later after we, or when we publish it, we just put a link in the comments so that people who are interested can just follow with the group. Maybe because I think everything we do for the well-being, or getting less worse for patients, I think it's just worth spreading mouth, and if it's helping one person, that's already good. Right?
Yeah, definitely. Yeah, and the housing setup. So they have. So they're still looking for collaborators. They announced the first set of technologies that are going to be within that house, and they did a ribbon cutting about a month ago in June. So they actually do have residents living there already. It's not a very big place. I mean, I think it has like maybe 80 residents total, and it can. They have about 10 right now. Yeah. But what they did have so far was not like, I think they could do with potentially more like biochemistry oriented approaches. They had one I had mentioned, I think when in our pre-discussion, that they have one company that is developing, or they have a toilet which has a sort of infrared sensor in there that can look at your stool, and then based on that, detect, okay, is there a certain amount of bloodiness or some other things in there? Yeah, so that we could send that information to some medical professional. And then there's another company, Cherish. They have a type of heart rate monitor and blood count monitor that is based off of tomography of some device in the room. So it's not something that you wear or strap to yourself. It's actually just a device that sits in the room, and then it can detect this. There are some of these tools that already exist that are in that area, but I think there still is potential for new tools that could actually help in, especially some of these patients with multiple cases or whatnot.
So cool. So before we go back to the deeper of QAA, because did you look, or where is the community thinking about sensors, quantum sensors, enhanced quantum sensors, or is it just not? Is this too far out? Because this is one of the aspects I think is also condensing that quantum and quantum 4 bio is going to play maybe a significant role in the not too far future, or is this too far out for you guys?
So this is something that we at IF are very interested in. We actually helped start the first Quantum Economic Development Consortium meeting on like biomedical sensing. My co-founder and chief strategy officer helped start that last summer with NIH and some of the companies that have sort of been developing in this area, such as Sandbox, AQ, and others.
Yeah.
So this is something that, so this is something that we're definitely like really very interested in terms of why we are very focused on quantum networking as a potential end state that we would like to see the industry work towards, rather than just like having quantum compute devices that exist. Because if we're able to network compute with sensors, especially for doing some types of new imaging modalities or whatnot, then the capabilities are much more interesting.
Yeah, that's true. And funny enough, I need to tell you this now, and I had a strange recording session with the Quantum Dragon from Brian Siegelbach's. I'm not sure if you know the Quantum Dragon. It's really
one, yeah.
And the Quantum Dragon mentioned that he wants to have quantum sensors and quantum computers working together with quantum networks, and I think that's exactly the future. So I think it's good that we are not the only ones to think about that.
Yeah, no, I think literally, like I was giving a presentation to Genentech actually recently. And in that I'm trying better to be like very clear about the difference between quantum chemistry, quantum information science, quantum devices, and then even within quantum devices, where does the quantum device become a quantum processing unit that can maybe do compute versus doing something else? So then in explaining the quantum device, I definitely try to stress that, okay, this is a device that can do some type of sensing that is at the quantum level. So you're dealing with quantum particles, or you have the ability to complete some type of quantum-like interactions. So, and then once you add another sort of processing unit on top of that, so once you actually add some type of, I mean, that's sort of been the way this has been done so far. I'm not saying this has to happen forever, but once you add an FPGA or some other type of compute layer on top of that, to then process what you would be sending to those to some quantum chip or to some quantum sensor, and then reading back those results and some meaning for that, then it becomes a quantum processing unit.
Yes.
Then once you have some actual algorithmic layer that you can then use on
If
top.
ever, yeah.
Then maybe you'll get to a quantum computer. So that's sort of the way, I think the things that we're, at least when we talk to biopharma, that's what we're trying to say, is that this multifaceted approach. There could be, for example, a quantum sensor. I think one thing that so there was the actual biomedical sensing workshop, but also one workshop that I participated in was frequency comb workshop that QADC put on last October. And that was interesting in that Dr. Scott Diddams from CU Boulder was there. I don't know if you're familiar with him or heard of his work.
Not yet, but I think I have read some stuff about frequency comps, but I'm not into the sophistication as some others have.
So his focus has been on frequency comb applications across the board. So just in biochemistry, but also in just other physics areas as well. And he was previously working at NIST, and then runs his own lab at University of Colorado Boulder. And so his interest now is looking at biological systems. So can you do some type of dual frequency comb setup such that you're able to, or maybe even triple frequency comb setup, such that you're able to do some type of cellular imaging in some way by looking at the differences in what that frequency, those frequency combs are measuring over some cellular image area. So that's something that he has recently been publishing a lot on, and there have been people in that sort of NIST CU Boulder community in Boulder, Colorado, that have been sort of working on this. I think one example that actually turned into a spin-out company is LongPath. I don't know if you've heard of the company LongPath before. So LongPath, they use this frequency comb method I think when they were developing it, it was not to the capability yet where you could do full cellular imaging. But what they found is you could at least use it for chromatography. You could do some type of chromatography with it. So they were using that for gas chromatography to detect methane leaks. So this is a product that they're selling to oil companies now, especially in Colorado, New Mexico, and Texas.
Why not?
detect oil leaks using quantum sensing. So that's definitely a set of conversations we've been wanting to have or having with biopharma, is like, okay, what kind of quantum sensing could exist? I think the other example, the last one I'll give here, and I'll try to also add these to the discussion afterwards too, so people can take a look. I don't know if you've heard of Peter Maurer at University of Chicago or his work before on molecular qubits.
On what?
Molecular qubits.
Yeah, I've heard of this, but I didn't follow. So I think this is definitely one thing we completely are forgetting at this moment when focusing on all these dead things like superconducting and ultra cold stuff. So I mean, my personal thing is, maybe I'm outing myself here, but why not? So definitely, I guess we are all in a quantum setting. There is definitely, I mean, we can go wild and say Hameroff Penrose believe that the OR model is, so the brain is just nothing more than a big system to manage quantum information, etc. But I think these sensors or color centers and specifically organic molecules, they should behave less bad than if you have to control stuff in nanokelvin, for example, or microkelvin. So you work with those guys, or?
Well, hopefully. I mean, so he is running one of the national. So there are a set of recently funded national quantum virtual labs and QVLs that were started here in the United States.
Okay.
So the goal being that they would create some type of virtual test bed that all of us, especially as startups and other companies in the United States, can use new technologies. But his specifically, so his was interesting because his project was University of Chicago working with MIT and the other Merck, not your Merck, US Merck, Merck Sharp Dohme. Yeah. So in how you could do some type of chemical sensing to be able to help in screening, so high throughput screening of some kind for drug discovery. And this builds on his previous work of developing this molecular qubit out of basically a version of the green fluorescent protein. So that was something that he was able to develop into some type of almost like a type of optical qubit, or sort of similar, actually probably more similar to like neutral atom qubits or whatnot that exist currently. So he has written some work on using this and developing this and how it can be this platform. He kind of mentions just towards the end, but that this platform could be used for variant analysis of some kind, because then you'd be able to look at the actual difference in measurement of your specific state to then be able to look at what type of variant could exist for that protein. So I found that part to be interesting and a little bit different in terms of
it's
use case.
Refreshing to have new use cases.
Yeah. But I think the bigger thing is the classical stuff has advanced a lot. I think definitely here in this area, the one tool that has been used a lot and people talk a lot about, and I think probably people you work with talk a lot about as well, is spatial omics. So tools like 10x Genomics' modeling and imaging tools. So there's sort of a belief that, okay, why would we need some of these better methods? But for example, with Scott Didham's frequency microcomb setup, that did not require using some type of protein to do some type of staining or anything like that. That was completely like just from imaging alone, they could do a very good analysis of, okay, here's like at least the macromolecular machinery that exists within the cell. Not going into very protein level depth, but at least where you could at least help with identifying what these proteins could be. Obviously, the cost is still not at a level where it's necessarily going to be in every lab yet. But that definitely is very compelling in terms of a lot of these existing methods classically do still require some type of staining, or you need to have some type of, and that obviously makes it difficult for especially transmembrane proteins, these ones we care a lot about for biology and immuno-oncology especially. So that's where the quantum methods are definitely becoming it. The use cases are starting to come out.
It is cool to share. I really like to hear this confirmed, because you pick up signals later today. I will have another interview with similar tendencies, and but I think we should come back one step, because I think you're passionate about this QAOA, and I like to joke about QAOA because QAOA is quite good if you have fully fault tolerant and very big and whatever machines. And my skeptic friends, they will listen in closely if you say you have found something with QAA that is help preparing protein docking or something like that. And I guess I've read this, or you told me that you outperform classical things. Can you? I think here's the chance to to explain.
So definitely there have been papers where they do claim this kind of thing, and it's not something that necessarily we've written about as explicitly in terms of QAOA alone. So, for example, recently, actually, last year, there was a team from, I believe, Tsinghua University in China. They had come up with this QAOA max cut that could be used for once you know where your binding pockets are. It could then be used for then identifying where those pharmacophores are interacting with each other. On those binding pockets, such that you could then identify which the maximally best set pharmacophore interaction out of some set of pharmacophore interactions,
Okay.
such that you could do some version of docking protein interactions. So then I think NVIDIA noticed this, and so they have a guide on the NVIDIA. I think like whatever their guidebook is for like using the CUDA-Q system.
Yes.
Here's how you can implement this. Pfizer picked up on this. So Pfizer wrote a paper on basically using that method, but then just doing it at a larger scale.
And this was just published today, isn't it? So I just read it in LinkedIn, I guess. Earlier today, I saw some graphs and some people were working, but okay, maybe this is the paper.
No, the preprint of this came out a couple months ago. But yeah, maybe they've like published it in a journal or something.
In a review journal, yeah.
Or it's been reshared. I know that the CTO of Pfizer did reshare it, saying, "Wow, look, we're doing quantum." Yeah. So there's interest. So there are people who do that, and they're claiming that okay, we're going to outperform or whatnot. Our use case and interest in QAOA, right? I say that all to say our interest in QAOA is very different, and our way in which we use it is very different. Mm-hmm. So our interest specifically, why we use QAOA is, I mentioned the client we have at UC Davis Medical Center. So her name is Dr. Angela Haxku. Her focus is on surfactant proteins. So these are proteins that I don't know if you're familiar with them very much. But I guess I'll explain for the audience.
Yes.
They basically stop your lungs from exploding when there's an interface between air and your cells, which are liquid. These surfactant proteins help to maintain the tension between that liquid interactions and also the air that exists, such that the air is not just colliding and destroying.
You don't
cells. So the surfactant proteins can be found on the outside of the alveolar cells in the lung, and therefore are part of your mucus. She's been interested in surfactant protein D for a very long time, especially because surfactant protein D was found to interact with influenza A and SARS-CoV-1. So she was sort of believing that, maybe there's some kind of interaction with SARS-CoV-2. So it was that I'm trying to understand that interaction without being able to do molecular dynamics, because at the time in 2020, most high performance computers were being used for doing other molecular dynamics for SARS-CoV-2. So it was at that time that we're like, okay, we have access to a Rigetti device, and we have this understanding of QAOA max cut that we could use towards protein pruning. And maybe that'll help with making the docking a little bit more straightforward. So specifically, what we wanted to do was not do the full docking with QAOA, but instead just help identify what are those atoms that are blocking meaningful binding sites. Because in this case with SARS-CoV-2, we really needed to do that because at that point there was not an understanding of where those binding pockets were. So we're like, okay, we need to come up with this understanding sort of ourselves. So we're like, okay, we'll use QAOA max cut to help identify those pockets, cut out those blocking atoms, such that we're left with this protein which just has the protein structure that just has these interacting sites that are meaningful. And then for each of the proteins, so this makes our protein docking problem much smaller. Now we can use existing docking methods like a Z-dock or a hex dock or whatnot to then see what these interactions are. So all I'd say, our interest was specifically on preparing for docking, not on the docking itself. The reason why is because that identifying the binding site, like I mentioned, is very hard to do if you have no chemical idea. If you people have characterized in ELISA or other types of assays what these interactions could be, right? So that's or through some type of molecular dynamics. So that's where we're like, okay, this could be like potentially useful. Now, in terms of the of course you have some problems with scalability of QAOA and on devices, but the scalability issues are actually like a little bit more, I think, interesting than I think most people are aware of. So classically, we have done some research, and we actually published this at American Physics Society earlier this year. where like if you scale the number of basically the way in which quantum approximate optimization algorithm works is you have the you have a Hamiltonian, so you have an energy function that describes in most cases some type of Ising interaction or some type of magnetic frustration of some kind, and then you have some type of cut Hamiltonian. So you have some other type of energy function that describes how to cut within that. And so you mix these two functions together using some unitary operation, so some type of matrix operation that combines these two. And then you are trying to solve this unitary operation on a quantum device, or because this is why it's sort of written this way. So that you could try to do this on a quantum device by using a quantum processing unit by doing trauterization. So you turn this Hamiltonian matrix into this set of this time evolution that occurs over some set of steps. So we had completed this poster on how the number of Trotter steps affects the result that you're getting.
Yes.
And if you run QAOA classically, you'll see that as you continue to increase the number of Trotter steps, you get a better result. So you get this nice curve where over time you get this better GTO as you continue to increase the number of steps. But on a quantum device, what was found just after we put out this poster, we saw that there was a benchmarking study completed by the Jülich supercomputing there in Germany, where they had benchmarked QAOA on the Quantinuum device, the IQM device, IonQ, as well as the Rigetti device and the IBM device. So they did superconducting, ion trap. So they tried different types of devices, and they found that actually you get a really good cut ratio initially, and then after just running a few Trotter steps on the device, and then you get really bad results right afterwards. So all that to say, so what is really interesting about QAOA for max cut specifically, and why, again, I guess why we're interested, sorry, going like a very long roundabout way, is so the maximum cut problem. So finding this maximum cut on a graph of where you can take the maximum number of vertices is, so algorithms that have been used to solve this maximum cut problem have been known to be non-deterministically polynomially complete, right, or NP complete. So we can count how many instances in which we can actually get to some polynomial or non-deterministically polynomial result. It's still not generally, we do not have some nice polynomial form that we can say, okay, given some n number of vertices, it'll take this n to the fourth amount of time, for example. But QAOA as an approach was found by Farhi, who is known as sort of like the father of QAOA, because he wrote about this in 2014. It's found to be a bounded quantum polynomial algorithm. So it can run in polynomial time, give it some level of bounds on a quantum circuit, right? So that means that, you may not be getting the same result as, say, Goemans-Williamson, which is the classical best-in-class method for solving maximum cut. That is, but that is an NP-complete method.
Yeah, so at least you get something.
Whereas QAOA is BQP. So so I guess it really comes down to, okay, I think where it comes down to is, do you care about solving max cut? Is it an important thing to solve for the problem you're interested in? And in our case, we're talking about large-scale proteins where we're trying to do some type of mathematical operation to find what are these interesting, or at least eliminate the blocking atoms that are blocking maybe some interesting binding sites where we don't have that kind of chemical understanding. So that's why we are using QAOA max cut, because it's a polynomial, or a bounded quantum polynomial algorithm for attempting this type of this type of problem, the maximum cut problem for proteins. So yeah, very different than I think what most people have been trying to do, which is trying to see, okay, can you take QAOA and somehow get some, because you can run it on a device, can you somehow get some advantage out of just like running it and seeing how to do some kind of problem with it, which obviously does not really work in all cases.
Yeah. There's a lot of passion. I see there's a lot of passion. I just look at the watch, and thanks for the listeners who are still. It's getting it. I like it, so I could hear to these things for hours. I just want to switch a bit gears.
Sure.
Talking about motivation, purpose, values. I think there's both of these. I have a bit the feeling play a huge part in your life. So you're one of the more. Is there a hack to bridge or something you can or want to share between environmental stuff that you're doing and quantum research and health innovation? I mean, we talked about it implicitly, but is there something you would let's go maybe a step further? Is there something you could recommend to either your younger self or being a mentor to other younger selves or other people who want to switch from being boring office stuff or to get inspired by working with nature and quantum?
Yeah, I think definitely. I think one thing that I do end up talking a lot with, especially other people who say, "Oh, this is something interesting. This is something that I want to do. This is what I'm interested in," is that there is definitely some meaningful economic value in doing this. I think a lot of people when they think about either the native plant stuff or what I do as full time with quantum algorithms would say, "Oh, this is." I've met people where they say, "This is a fun side project. This is really interesting, but this is not something that I can necessarily I think is necessarily like a full time thing." But I think that's fundamentally an incorrect understanding of that in terms of of course the quantum approaches for these kind of biopharma applications. I mean, this is something that is still very much at even 40 plus years after QSAR and some of these methods have already existed. Okay. It's still very much at the ground level. There's still like a lot of understanding and tools that can be developed that can really help in better providing a way through which quantum chemistry and now quantum algorithms can actually provide some meaningful value in terms of solving some of these really, really hard problems wherein we don't know, like, I think the hardest problems are the ones where we don't even know the complexity, right? Yes. We can't even formulate an algorithm to know a complexity of it. So these are these problems that do exist in biopharma still, especially when we're talking about transition metal interactions, or especially when we're talking about these transmembrane proteins, especially when we're talking about microbiome systems that we don't have any information for, like especially in the ocean, deep in the ocean or whatnot. That's where that would be my biggest motivation, and also just understand what makes the most sense for you. I think one thing obviously you'll notice about IF is we have not raised yet, and it's been a conscious decision because we have been very fortunate in that we've been able to generate revenue from clients, and I think definitely my experience with a number of different companies, especially like my first project started out was with IBM, and then ended up working at Deloitte and whatnot. It's really helped with bolstering that. But I would say it's unfortunately been a little bit of necessity because we just haven't met the right investor or people where we feel like, okay, they have the same mission as us. They really see that there's a long-term goal that can be achieved with a quantum, where we will have a quantum network between compute and sensors, and that we will be able to then have biopharma use cases. We have not been able to meet people who really have that vision. We hope that's changing now.
Yes,
Maybe.
yes. So just a call to the investors who are listening to this. We're talking about the industry transformation. I will have to do some calls later. So just no, I think it's really so.
Yeah.
It's early days, but it's not too early. And I think you just confirmed we are in this a kind of an industry transformation. I mean, even Nvidia is saying we are getting ready. So even though they don't produce quantum hardware, but I think the industry is waking up. IBM is producing, everyone, Quantinuum, everyone is just pushing, and we see the first big mergers, billion mergers. So I think before we jump to the quantum pool, because I think we will have. I started a quantum computer earlier before recording, so it chose already a cool question. But so this, can you have one statement on readiness, quantum computing readiness? I mean, would be your take? So one month, 2 years, 25 years to run your algorithms on hardware, or also how is the perception, the readiness? I think you said sometimes the biopharma industry is not yet really ready to understand what quantum could add to the table. So on a level of maturity, where are we?
Yeah. So the thing is, in terms of a quantum processing unit that can take an algorithm, a quantum algorithm, and run it based on exactly how one should expect, based on numerical simulation for that quantum algorithm to then provide some solution. Of course, that's something that it remains to be seen, but I think maybe the approach in which we have been looking at this so far as an industry or really an industrial niche has been maybe not the most correct, and I think it seems like it's changing for better. So like I mentioned in the case of QAOA, for example, there's a distinct difference between what runs on the device and what you would be getting from some type of numerical simulation, chiefly because there's natural intrinsic Hamiltonian that exists on the device itself, right? And that is trying to embed some other problem that you're working with. So of course I think basically all that to say that there are certain advantages that maybe a quantum device will have, and we have been very fortunate that we've worked with. We were part of Xanadu's quantum sandbox. I mean, we've worked with IBM, so I've worked on I think pretty much all modalities that exist so far to say okay, there are certain distinct maybe wall clock time advantages or state space advantages that exist. I mean, the state space one I think is the one that everyone talks about. Once you get beyond 30 qubits, you're talking about many terabytes of output if you actually want the whole output and not just some.
If you find the correct encoding, yes.
So yeah, I think that definitely the bigger problem is not are we there yet in terms of getting the algorithms there. I think that's sort of the way it's framed, but rather what are the cases in which a quantum algorithm may be running in a quantum simulator is providing some advantage versus a quantum algorithm that's running on some type of actual compute device, and how can we best develop these devices to be more like ASICs that can do very specific problems very well. I think the other thing that's kind of crazy is it was one thing, I think, when we first were discussing this in 2020, 2021, and then at the start of when QFarm was really running, that what's the horizon for quantum? Because in general, for biopharma, because biopharma in general has been a very the way people keep telling me is it's a very conservative industry. They want to view technology from the step-by-step method.
Yeah, don't make errors, yes.
Yeah, but I kind of don't feel that way, because I think now, especially with the application of LLMs by most in biopharma and biopharma companies saying, yeah, we're going to use the LLM for doing all of our drug discovery. It's going to be amazing. And then LLM being, in many cases, probabilistic and even worse in terms of outcomes than some of the quantum algorithms, and in terms of determinism of actually knowing some type of computational complexity to say, okay, this is how how some actual algorithm will scale to get some result. I don't understand why the approach still that quantum is way out when LLMs are somehow fine, even though the complexity is unknown for a lot of these use cases, and it still is yet to be developed into a way in which you can properly do some kind of source analysis or whatnot and see where you're getting these results from these LLMs. So I think in context, quantum is actually in many ways a little bit more established, and there exists known complexities for algorithms in quantum that don't exist for LLMs and machine learning. And so I that's the sort of message that I've been trying to talk to people about.
This is cool. I mean, in principle, we could even say, let's do an LLM simulator, and then instead of deploying it as an LLM, you just try to get the error function of the LLM, and they're good. So I start the quantum random because I have a nice sound for that. Just wait, I'm starting it. I'm starting again. Good. I have one, and funny enough, it ran again on IBM. Which one? Brisbane. Using 7 qubits for my algorithm because I have not as many questions as we need the full processor. The question index is number 42, just for those guys who who take minutes, and therefore it means it's question number 43, and it reads now. What's your favorite way to stay curious in daily life? I mean, maybe you're a good person to answer that.
What's a good way to stay curious? Oof. I think definitely spending time in nature for sure. It's
not scripted. It's really random.
Yeah, definitely. I think that one's the main one, especially. If I can visit places that have these native plants and whatnot that are not monocultures, those are the most interesting, because then you'll come across some maybe some fungi, or you'll come across some type of plant and say, wow, what is this? How is this able to grow in this way? So that definitely keeps me always curious and wanting to learn more about the chemistry and then maybe even the applied biochemistry that could be gain from that. I know, very very boring in the long scheme of things, but for me that definitely rejuvenates me.
We do the interview, yes.
Makes me interested. But how about for you, Thomas? Way which you stay curious about things.
You give back the question. So good point. So, I think I take my energy also from nature, if ever it is, but also very much from these kinds of interactions. Because dialogues, I always try to do my very best, and I know that I'm failing very often, but it's active listening. to really listen and not anticipate what the other guy's saying. But I think this is really good, and also looking at the broad diversity. So looking at people who are not sitting in the office but doing arts or having mental disabilities is super inspiring to talk with and work with those guys. So that's, I think, for me, really curious to learn how people with mental disabilities see the world and what we can learn from them. So this is my inspiration.
Yeah. I was learning. I was seeing your you run a kitchen, right? Or you help.
Yes. So let's do marketing for the Glückskeks. Yeah. So my son, he has he was born with Down syndrome, and they have now it's not a club. It's not. I mean, it's a kind of an association of some 10 people from age 16 to age 21 or age 22. And they come together, they have fun, they cook, they produce oil, they produce what have you. And we are running now this every second Sunday, we are running a coffee in the home for elderly, which means those people have time. And because sometimes it really takes time, but it's really a very, very social interaction, and it's amazing to see the way if you live not only with the brain but with your heart. So how this changes the way you see things in life and how this then ripples out to the universe. So this is really cool. I mean, it's a very small step. It's every second Sunday for 4 hours. But I think that's it, what we're doing. So it's yeah. Yeah,
Well,
thanks for asking. Now for the sake of the planned thread of the series, every guest has also the opportunity to provide his or her own question, and then post this question and answer it yourself. So just go.
yeah. So my question, which I want to hear your answer for as well, is if you could, using whatever latest technological advances we have, what therapeutic area do you feel is an unmet area, and why would you feel that we should solve this specific area, or like what specific disease would you be interested in solving and looking at too? And I guess I'll answer first. For me, it would definitely be I think the like ovarian cancer is definitely one that I think is not it's getting better understood, but it's still not like very well understood, especially given its impact on reproductive health and whatnot. And I think there are a lot of what's interesting in looking at the protein interaction networks and then the actual protein interactions within this network, is that there are a lot of overlapping proteins that exist between the sort of so that reproductive area, as well as in lung health. So actually there has been some findings that surfactant proteins have actually been found in and released in vaginal tissue and whatnot.
Okay. Wow.
Some of the work from one of the research centers focused on surfactant proteins. So I think that's very, very fascinating, and that's an area in which developing these sort of biologics can either be the platform for some small molecule drug, or they can themselves be the molecule that's interacting with some of the diseased proteins that are part of this cancer. That would be like a very interesting area to look into, and I think the impact would obviously be tremendous, considering that there's such a lack of understanding of women's health and reproductive health, that like I think these kind of new technological methods where we can do better sensing, we can do better imaging, and we can do it in a potentially less invasive, more healthy way. That's where I think like I hope the industry is going, and also can help with that in some way.
Cool. So I answer my part, and then I give you the last word. Sure. I don't want to close with my own words. So if I got you right, so what is the disease or whatever I would like to cure? So I think it's ignorance. Unfortunately, it's ignorance, human ignorance, and the putting the ego in front of everything. I'm not sure if there's a medical cure for it, but I think what I would hope that we discover is also maybe with more sophisticated insight how quantum really works, is that eventually, like, we are all one. So that's why I call this Entangled Health. But not really kidding. The thing I think would be cool, also from an industrial perspective, and you can market this, is to have this kind of an enlightenment enhancer. So I was rather thinking about imagine what it would be like that if everybody who is not in a same fortunate situation that I was, which means being loved by his or her parents, this gives you a trust, and then you can just work differently than if you're afraid from being born. You're being afraid, and you need to do things right, and you're just not feeling this love. And just imagine we find a way to, it's a brain wave, so what is it, to measure this intrinsic gene pattern / brain pattern that gives you this trust. I mean, and then you just put the cap to the other guy, download and press enter. And then bong, you have this feeling, whoa, this is how it feels. So maybe I need to be less aggressive. So I don't know. So this is my 25-year project, which will get us maybe to world peace. But I don't know. So that's it. But we need, I think, all different perspectives to measure it. What is it that makes people? So this fundamental trust. Can you give it back once it not when it was not there, or I don't know? Is everybody born to make their own history? And for good reasons, there are these people who are not so good for the world peace. So is this for a reason? I think yes, but could we stop it with this? And do we just wait for this enhancer to stop it? I don't know. This is.
So you want some type of oxytocin, or I guess you're looking at something beyond oxytocin, potentially.
Well, yeah, ayahuasca for everyone, but I don't know. But yeah, if I would knew, I would really stop what I'm doing and then just put my full energy there. But I don't know how it would work. So therefore, I have to do this podcast to find out. So this was really a pleasure, Samarth. Do you have something as a closing statement?
Yeah, I would say my closing statement, I think based on some of the words that you said, is that please don't live your life in ignorance. Read papers, reach out to human beings about problems you see, and don't trust your LLM blindly.
So, dear friends, thanks for listening and entangling to the Entangled Health podcast. Feel free to like it, share it, subscribe on your favorite platform, and if you are inspired, join our guest and submit your question for future guests. Join the conversation either via sending an email, following us on LinkedIn, commenting under the episode. Stay curious, stay connected, and keep exploring the unseen with Entangled Health.